one parent or two. This is the fact that determines whether the reproduction will be asexual reproduction or sexual reproduction. We have seen in our previous videos about asexual reproduction. In asexual reproduction, there is no involvement of gametes. Only single parent is involved. There is no zygote, no embryo, but there is progeny only from a single parent. And the progeny is identical to the parent. So we have to move on to sexual reproduction now. So before you go on to sexual reproduction, I just wanted you to have a comparison or an understanding of how different asexual reproduction and sexual reproduction is. The points to be compared, the facts to be compared are the complication of the process. Asexual reproduction is not much of a complicated process. It is a simple process. One cell divides into two, or a broken fragment develops into a new organism, or a spore germinates into a new plant. Like in the case of vegetative, there are stems rising from tubers. So the process is simple. Whereas in sexual reproduction, the entire process is a complicated process. Another factor to be considered is the rate at which this process is taking place. So asexual reproduction occurs fast. So the process takes very less time, and within the less time we see more number of progenies. Whereas in sexual reproduction, the time taken is a lot of time. Much time is taken for the progeny to grow and to be given out. So this is one factor to be considered. And when you look at the number of progenies, asexual reproduction, the number of progenies are more, definitely more. Okay, more organisms, more plants. One eye giving many stems on the potato tuber, you have seen. So like that, the number of progeny is more. Whereas in sexual reproduction, you cannot expect that many number of progeny. In human reproductive system, it is always one progeny during one cycle. The last but not the least important difference is the variations that are produced. In asexual reproduction, we do not have any variations. The progeny that is coming out is exactly identical to the parent. So what the parent is, the progeny is the same. Whereas in sexual reproduction, there is something called genetic recombination taking place within chromosomes. So there is the characteristics from the male gamete and some from the female gamete are contributed, and thereby the progeny is a new individual. It is resembling the parent, but it is never identical to the parent. The progeny that is coming out of sexual reproduction resembles the parent, but is never identical to the parent. But in asexual reproduction, you see that the progeny is always identical to the parent.
Asexual reproduction has its own advantages and disadvantages. The advantages of asexual reproduction are: the population can increase rapidly when conditions are favorable. Only one parent is needed. It is time and energy efficient as the process does not require a mate. It is faster than sexual reproduction. The disadvantages are that there is no genetic variation since progeny is identical to parent. It makes adaptation to environmental changes a more difficult process. The entire community of organisms that are genetically identical might not survive the changes. A disease may affect all individuals. Asexual reproduction may result in overcrowding, thereby leading to competition and shortage of resources. What is the advantage of sexual reproduction then? The process produces genetic variation in the offspring. The species can adapt to new environments, which gives them survival advantage. If there is any disease, the disease is less likely to affect all the individuals. The disadvantage of sexual reproduction is that time and energy is required to find a mate. That's all. So, looking at all these factors, sexual mode of reproduction is considered to be better than the asexual mode of reproduction. Let us now move on to study sexual reproduction. Sexual reproduction is the formation of a new individual following the union of two gametes. Fusion of gametes is by a process called fertilization. Fertilization may occur outside the body of the female organism, which is called external fertilization, and it can be seen in fish and amphibians. The female lays a vast number of eggs into the water, and the male releases sperms on them. Fertilization is purely by chance. Fertilization may occur inside the female organism, as seen in land animals. This is called internal fertilization. To understand sexual reproduction in animals, we study about the human reproductive system. Under this, we will be studying about the male reproductive system, parts and their functions, structure of the sperm, the female reproductive system, parts and functions, what happens after fertilization, and about childbirth. First, let us look into the male reproductive system. The parts of the male reproductive system are a pair of testes, Vasa efferentia, a pair of epididymis, Vasa differentia, a pair of seminal vesicles, ejaculatory duct, prostate gland, a pair of Cowper's gland, and the urethra. First, let us understand the testes. The testes are a pair of oval sperm producing organs in the male reproductive system. The testes are known as gonads. They are located outside the abdominal cavity within a sac like structure called scrotum. Within the testes, there are finely coiled tubes called seminiferous tubules. These tubules are lined with a layer of cells which develop into sperm. This happens from puberty through old age. The sperm cell is the male gamete. The process by which this takes place is called spermatogenesis. Spermatogenesis is by meiosis. Main function of the testes is to produce and store sperms. They are also necessary for secreting testosterone, which is a male hormone. The next part is the Vasa efferentia. The highly coiled convoluted tubules that lead from the testes and form the head of the epidermis is the Vasa efferentia. The next part is the epididymis. The Vasa efferentia are highly coiled tubes that open into the epididymis. They are present on the posterior side, that is the back side of the testis. There are one on each side. It stores and transports sperms that are produced in the testis. Next is a Vasa differentia. These are muscular tubes that transport sperms from epididymis to urethra. Please make note of the difference between Vasa differentia and Vasa efferentia. The Vasa differentia arise from the epididymis, whereas the Vasa efferentia arises from the testis. Vasa differentia carries sperms from epididymis to urethra. Vasa efferentia transfers sperms from testis to epididymis. The Vasa differentia are two in number, whereas the Vasa efferentia are many in number. The next important part is the seminal vesicles. These are vesicular glands. There are a pair of vesicular glands, coiled tubular glands they are, and they lie behind the urinary bladder. These are accessory glands of the male reproductive system. They produce seminal fluid that will later contribute to semen. The next part is the ejaculatory duct. Each ejaculatory duct is formed by the union of vas deferens with the duct of seminal vesicles. They pass through the prostate and open into the urethra. The prostate gland. It is a walnut sized gland located between the bladder and the penis. It is just in front of the rectum. Rectum means the end of the large intestine. Urethra runs through the center of the prostate from bladder to the penis, letting urine flow out of the body. Prostate secretes fluid that nourishes and protects the sperm. Cowper's gland. It is also called bulbourethral gland. It is a pea shaped gland located beneath the prostate gland. They add fluids to semen. They produce a lubricating mucus secretion, helping in lubricating the urethra. The next part is the urethra. It is a tube that extends from the urinary bladder to the outside of the body. It also connects to the vas deferens in males for ejaculation of sperms. There is a sphincter at the upper end helping in controlling urine in the bladder. The function of the ureter is to allow passage of urine outside for excretion. In men, it serves an additional purpose of transporting sperms in semen. What is semen? Semen is also called seminal fluid. It originates from seminal vesicle. The fluid is also contributed by prostate gland and the Cowper's gland. The process that results in discharge of semen is called ejaculation. Semen carries the sperms capable of fertilizing eggs in the female. Since we have understood all the parts, we can now see how the spermatozoa pass through the male reproductive system. Next, let us understand the structure of the sperm. Sperm is a microscopic motile structure. Motile means highly movable. It is composed of a head, neck, a middle piece, and a tail. The entire body is covered by plasma membrane. The sperm head contains an elongated haploid nucleus. Haploid means it will have half the number of chromosomes. The anterior portion of the head is covered by a cap-like structure called acrosome. The acrosome helps in entering into the ovum, helps in projection into the ovum. The middle piece contains numerous mitochondria which produce energy for the sperm motility, which is needed for fertilization. The head and the middle piece are attached by a small neck. The tail helps the sperm cell to swim in order to reach the egg cell. Now let us understand the female reproductive system. The parts of the female reproductive system are a pair of ovaries, a pair of fallopian tubes, the uterus, and the vagina. Ovaries. These are the female gonads that produce ovum. They are oval shaped. They are located on either side of the uterus. They are held in place by ligaments attached to the uterus. Inside the ovary, there are follicles. The follicles contain immature eggs. At the beginning of each menstrual cycle, several follicles enlarge and migrate to the outer surface of the ovary. One follicle is selected for further growth and maturation. The mature follicle is called Graafian follicle. The fluid in the Graafian follicle secretes ovarian hormones. It also contains the oocyte. The mature follicle, under the influence of female hormones, ruptures and releases the egg. This process is called ovulation. The released egg passes into the fallopian tubes. The fallopian tubes are also known as uterine tubes or salpinges. They extend from uterus to ovaries. The fallopian tube allows passage of an egg from the ovary to the uterus. Here, in the fallopian tube, the egg is able to fertilize with the sperm. The uterine tubes transport the fertilized egg to the uterus for implantation. What is oogenesis? It starts with the oogonium. This cell undergoes mitosis to increase in number. The cell starting meiosis is called the primary oocyte. This cell will begin the first meiotic division but is arrested in the first prophase stage at birth. During adolescence, hormones cause development of a number of follicles in the ovary. This results in the primary oocyte finishing first meiotic division. The cell divides unequally with most cellular material and or organelles going into one cell. This will be called the secondary oocyte. A small amount of cytoplasm and one set of chromosomes go to the other cell. This second cell is called the polar body. It usually dies. A second meiotic arrest occurs, but now it is at the metaphase stage. The secondary oocyte is the one which is released at ovulation. It will travel towards the fallopian tubes. If secondary oocyte is fertilized, it continues through meiosis II, completing meiosis, producing the second polar body and a fertilized egg containing 46 chromosomes, half of which are coming from the sperm. The result is a zygote which undergoes mitotic division while traveling down the fallopian tube. The next part is the uterus. It is a hollow, muscular, pear-shaped organ located in the pelvis between the bladder and the rectum. The lower end of the uterus is the cervix. It opens into the vagina. The upper end is called fundus and is connected to fallopian tubes. The uterus has an inner lining called endometrium. It is an epithelial layer with mucus. The endometrium changes in thickness during the menstrual cycle. It is the thickest during ovulation. If the egg is fertilized, it attaches to the endometrial wall of the uterus and starts developing. If the egg is unfertilized, the endometrial wall sheds its outer layer of cells, the egg, and excess tissue in a process called menstrual bleeding. Endometrium helps to keep the fertilized egg implanted and provides suitable environment for growth. What happens after fertilization? The newly fertilized cell is called a zygote. The zygote moves down through the fallopian tubes into the uterus. In the uterus, the cells continue to divide, becoming a hollow ball of cells called the blastocyst. The blastocyst attaches to the wall of the uterus. This process is called implantation. The wall of the blastocyst is thick in one area. The inner cells in the thickened area develop into the embryo. The outer cells burrow into the wall of the uterus and develop into placenta. The placenta produces several hormones that help in maintaining pregnancy. It also carries oxygen and nutrients from mother to fetus and waste materials from fetus to mother. Some cells from the placenta develop into an outer layer called chorion around the developing blastocyst. The chorion is the membrane between mother and fetus. It is the outermost membrane. It contributes to the formation of placenta. The chorion develops chorionic villi, which are finger-like projections that pass through the endometrium and eventually connect with the mother's blood vessels. Other cells develop into an inner layer called amnion, in which forms the amniotic sac. When the sac is formed, the blastocyst is considered an embryo. The amniotic sac is filled with a clear liquid called amniotic fluid. This expands to envelop the developing embryo. The embryo is thus protected. The allantois is a hollow sac-like structure with clear fluid that forms a part of the embryo. It eventually forms the umbilical cord. It lies between the amnion, which is the innermost layer, and the chorion, which is the outermost layer. It helps in exchange of gases and handles liquid waste from the developing embryo. The umbilical cord develops from the allantois and yolk sac, and joins the placenta. It transfers material to and from the maternal blood. There is also a yolk sac with fluid, but it has no specific function in placental mammals. As pregnancy progresses, the fetus with additional characters will grow and the uterus will increase in diameter. At about 9th month, the head of the fetus will turn down towards the opening of the uterus. Usually the head comes out first, but sometimes the feet may also come out first, but this type of delivery will be a difficult delivery. Childbirth will begin when the muscle layers of the uterus start to contract and relax rhythmically. These are called labor pains. The muscular activity of the uterus is strong enough to move the baby slowly towards the vagina, which is the outer canal of the reproductive tract. At this stage, the amnion around the baby breaks and the fluid contents are released. So this is what is called labor. The contractions of the muscles become stronger and more frequent to propel the baby out through the vagina, and then the baby is delivered.